Applications of continuous culture. In industrial use, it is used in the production of therapeutic pharmaceuticals, antibiotics, ethanol production, and fermented food products such as cheese. Then in research field, it is used to collect data to be used in the creation of mathematical models of growth for specific cells or specific organisms, and analysis of biological processes in microorganisms, and to study biofilm formation in Pseudomonas aeruginosa. Next, in biological waste treatment and cell propagation. Applications of batch culture. It is used in the yeast cell production, in which sugar was added periodically during the course of fermentation to maintain a low sugar concentration to suppress alcohol formation. In penicillin production, where in penicillin fermentation, in which the energy source will be glucose and precursors is phenylacetic acid, were added periodically during the course of fermentation to improve penicillin production. Let's move on to microbial growth. Microbial growth refers to the increasing number of cells, not to the changes in the size of cells. What is generation time? The time required for a cell to divide or the population to double is called generation time or doubling time. There are 4 different growth phases. They are lag phase, logarithmic phase or log or exponential phase, we say, stationary phase and death phase. So let us know about lag phase. Lag phase is the first phase observed in your growth curve. It is characterized by no increase in cell number, but here the cells are actively metabolizing in preparation for cell division. After the inoculation of microorganisms or bacterial culture into the medium, immediate phase, that we'll call it as lag phase. Second one is exponential phase or log phase. In this phase, the cells grow most rapidly. Rapid multiplication of the inoculated cells you can see in the exponential phase or log phase. They grow most rapidly, doubling at a fairly constant rate. Primary metabolites will be produced in this phase. Next we have stationary phase. Here the number of viable cells is equal to the number of dead cells. The factors that cause cells to enter stationary phase are related to changes in the environment, typically caused by high cell density, depletion of nutrients, and accumulation of waste products. Secondary metabolites are produced in this phase. Next we have death phase. Here the number of viable cells decreases exponentially, therefore cells die quickly. So this is the last phase. This will call it as death phase or decline phase. Here is a graph showing growth curve, plotted number of bacteria on x-axis and time on y-axis. Here you can see the growth curve showing 4 different phases, that is lag phase, exponential phase or log phase, stationary phase and death phase.